All right, welcome to our next lesson on translation. So, finishing up protein synthesis with one of my favorite puns, the pirate ribosome. So, let's talk about what translation is. Translation is the process in a cell that's going to make proteins. It is the final process of protein synthesis. In fancy terms, you can say it is the process that generates a polypeptide chain. Since a polypeptide chain is a protein, it happens on ribosomes, but the ribosomes are located in different locations depending on whether you're looking at a prokaryote or a eukaryote. So prokaryotes don't have a nucleus versus cytoplasm boundary. So all of the ribosomes are free floating in the cytoplasm, which is where protein synthesis takes place. In contrast, in eukaryotes, you can have the ribosomes attached to either the endoplasmic reticulum, like what you see in this part of the diagram, or they can be free floating in the cytoplasm. Now, any given ribosome can either be, a ribosome does not have to permanently stay attached to the ER or permanently free in the cytoplasm. It can change its location depending on the mRNA transcript. So let's talk about ribosomes real quick. They are a tiny organelle or a very large molecule that is a complex of ribosomal RNA as well as proteins. And then, as I said on the previous slide, depending on some of the information on the mRNA transcript, which can determine where the final product of the protein is going to end up, the ribosome can either be attached to the rough ER or it can be free floating. There are two parts of the ribosome, a large subunit and a small subunit, which come together around an mRNA transcript. And there are three tRNA binding sites. I'm not going to talk in detail about them. I'm not really going to name them, and you are not responsible for knowing any details about them on the AP test. But it is helpful to know that you have these three sites because as a ribosome moves along an mRNA molecule, you're going to have tRNAs come in. So they're going to come into this site first. As the ribosome moves along the mRNA, the tRNA that was originally in that first site will move over to the second site. And then once it has given up its amino acid, it's going to move into this final site, which will then kick that used tRNA out. So first, let's talk about a little bit more detail about how genetic information is coded for in nucleic acids. The nucleic acids, DNA and RNA, are read three nitrogen bases, three nucleotides at a time in units called codons. Now, codons are not separated by any physical structure. That's simply the reading frame of the tRNAs and the ribosomes. Each particular codon is going to correspond to a specific amino acid. So, for example, here in this mRNA, you see the codon UUC is going to correspond to the amino acid phenylalanine. So, when you have UUC in an mRNA transcript, the corresponding amino acid that's going to be put into the protein is phenylalanine. Now, each amino acid, and there are about 20 that are regularly occurring in organisms, has a three-letter abbreviation and a single-letter abbreviation. You do not need to memorize any of these, and you don't need to memorize the names of the amino acids. You also will not need to memorize which mRNA codon goes with which amino acid. Let's talk, before we get into that, a little more about tRNA structure. So tRNAs can be represented in various different ways diagrammatically. Here, what we see, this is a more realistic 3D structure that shows you how the RNA molecule folds up. And this is a more kind of diagrammatic representation that's just going to show you the structures. The important parts of a tRNA for our purposes are the amino acid attachment site, which is where an amino acid is going to attach, as well as the anticodon. So depending on the anticodon that it has on one end, it will have a particular amino acid attached to the other end. And this match between a particular anticodon, which is going to be complementary to a specific mRNA codon, and the amino acid that's attached to the other end is going to be the basis of the genetic code in organisms. And this tRNA, once it gives up its amino acid, can be reused over and over and over again. So let's talk about the genetic code. Each codon represents a particular amino acid, and you can use the genetic code chart, which we're going to look at on the next couple of slides, to figure out which amino acid goes with which particular codon. There are about 64 possible codons that can occur in mRNA, and there are about 20 amino acids that are naturally occurring. So because of this, there are some codons that will code for the same amino acid. So some amino acids can be coded for by more than one codon. One amino acid, for example, has like 6 different codons that code for it. And there are other amino acids that are only coded for by a single codon. It just depends. This is called redundancy, and it's an important feature of the genetic code. What ends up happening is, since you can have these multiple codons code for the same amino acid, sometimes if you have a single mutation, a nucleotide mutation, or a mutation in one single nucleotide, that can change your DNA sequence and therefore your mRNA sequence, but still not end up changing the amino acid sequence, which then makes sure that a mutation that happens does not necessarily cause harm to the organism that has that mutation. Now, nearly all organisms use the exact same genetic code, and this is evidence for common ancestry of all living things. What this means is that the genetic code must have been established in the ancestor of all living things. There are very few exceptions to the genetic code, and honestly, they're found in a couple of different prokaryotes, and they may not even be evidence of this genetic code being different in those organisms from the beginning. Honestly, they're probably a modification of that exact same genetic code that is found in most organisms. So let's take a look at the genetic code here. This is a codon chart. There are 2 different versions. This is probably going to be the easiest version for most people to read. And the other codon chart, which is here, is probably a little harder for most people to read, but I will say this was I think the original codon chart back in the day, in the dark ages when I was a high school student. This was the only one that was available. So let's take a look at this codon chart. You basically start reading it from the center and you work your way outwards, just like these arrows show. Now, in the codon charts you'll find on the AP test, you'll always be provided with it when you need it for a question. You're not going to have these arrows, so you always need to remember if you have the circular codon chart, which I will admit I have not seen show up on the AP test, then you're going to need to read from the center and work your way outwards. So let's say that our codon that we're looking at is AUG. We would start from the center. And once we see our letter that we're looking at for the first base in the codon, we start from there and then we work our way out. So this is A. The second base is U, so we would work our way out. And once we see our first base, we're restricted to this quadrant, this quarter of the circle. So we need to work our way out, and we see the U is here. So now we are restricted to this section, which is all of these codons, AUG, AUA, AUC, AUU, will be found outward from here. Then our third base is a G, so we would look and find that our third base right here is the G, which means that this is our amino acid. Now, this is one of the only amino acids and codons that you're really responsible for knowing. AUG is what's called the start codon. Every single mRNA transcript starts with AUG, which means every protein, when it's originally produced, does start with a methionine, or MET. Now, if we look further, we see some other codons. Let's say we're looking at GUG. We would start here at the center with a G. We would go outward to a U, and then we would go outward again to G, which means GUG stands for valine. Notice all of these codons, GUG, GUA, GUC, and GUU, all are going to correspond to the amino acid valine. So that's that redundancy that I talked about in the genetic code. If we look and we see UAA as another codon. We see UAA. Notice on this one, on this chart, it's represented by just a square, but technically that's a stop codon, and there are 3 different stop codons. What that means is that doesn't correspond to a particular amino acid. That's just going to signal the end of translation. So let's look at this with this chart now. This chart I have found students tend to find a little harder to deal with, but it does have its advantages. For example, you can see that these 6 codons right here all code for the same amino acid. Leucine tends to have the most codons coding for it, whereas methionine, the start codon, and tryptophan only have 1 codon each. So the way we read this chart is we start with our first letter. So let's say we were looking at our start codon again, AUG. Our first letter here is an A. That means we are only going to look within this box. Notice that every single codon in this box starts with A. Then we're going to look for our second base, our second letter, which is U. So every single codon in this column is going to have U as the second letter, and everything in this particular box right here starts with AU. At this point, if you have this type of codon chart that has all of the basically has the codons listed by amino acids, by RNA sequence, you can just look in the box and find your corresponding codon, AUG. Otherwise, if you don't have that, some versions of the codon chart are going to need you to look at the third letter, which in this case would be G, and that would tell you AUG stands for methionine. If we look at a different codon, let's say CAU, that would require us to look for our first letter, C, our second letter, A, and that would be in this box. And CAU, our third letter, means this would be histidine. If you need to, go ahead and go over this part of the video extra times. If you need to see how to read a codon chart like this, if you need extra help, come see me in class. But now let's talk about the actual process of translation. So translation has 3 steps. The first step is initiation, the beginning. The rRNA in the ribosome is going to interact with the mRNA at the start codon, which always codes for the amino acid methionine. And then the ribosome is going to assemble around the mRNA at that start codon. And like I said, all mRNA transcripts always begin with AUG as the start codons. So all polypeptides originally begin with the methionine at the beginning, but that can be removed as part of what's called post-translational modification to change up the protein structure. The next part of translation is called elongation. This takes the longest time, and this will proceed as long as it needs to until you'll reach the end of the mRNA transcript. So let's look at elongation. So this is elongation. What happens is a tRNA is going to basically come into this entry site. It has a particular amino acid attached to it based on the anticodon that is complementary to the next mRNA codon that's exposed in this ribosome. Then what's going to happen is the new tRNA comes in, and any polypeptide chain that was attached to the previous tRNA that was already sitting in the ribosome is going to be transferred over and attached with a peptide bond to the amino acid in the new incoming tRNA. Once that happens, the ribosome shifts over to the next mRNA codon, and the tRNA that no longer has an amino acid attached to it then gets shifted over to that exit slot, and once it's shifted over to that exit slot, it gets kicked out. And then that tRNA can then be taken up by other enzymes and have the appropriate amino acid attached to it. Now we have this exposed entry site, which has a new mRNA codon exposed, and you can have a new tRNA come in with its matching amino acid. The polypeptide chain gets transferred over to that new amino acid, and then you end up having this sequence repeat over and over again until you end up reaching the end of the mRNA transcript with the stop codon, at which point you get to the next step. I will try to find some good videos that show this process in action and add them in the description. The final step is termination. So once you reach a stop codon, instead of a tRNA that comes into that open mRNA, not mRNA, sorry, ribosomal slot, you're going to have a release factor enter. It's not a tRNA. When that enters, it's basically going to trigger this whole complex to fall apart. So there will be no new amino acid for that polypeptide chain to be transferred to. It's going to be released and be a free polypeptide. The large and small subunit of the ribosome are going to basically fall apart, and they're going to be released, as well as the newly synthesized polypeptide or protein and the mRNA, which can then be translated as many times as needed. So, just a cool side note that I don't think is required learning for the AP test, but I always thought it was cool when I learned about this. So, you can actually have multiple ribosomes reading the same mRNA at the same time. This forms a structure called a polysome, which then speeds up the rate of protein production. So, in this diagram in A, what you're seeing is this whole transcription and translation process happening in a prokaryote, where it can happen at the same time. So here you see a DNA strand, you see an mRNA that's being produced from it, and you see ribosomes reading along that mRNA strand at the same time. On B, what you're seeing is a little more detail where you have kind of more of a close-up, and you can see this is an mRNA strand where this arrow is the 5' end where you start reading. So the direction of translation would be this way. And then you see multiple ribosomes along this mRNA, and you can even see the polypeptides that are coming out of the ribosome. And then, just like we had post-transcriptional modifications to modify the pre-mRNA, you actually can have modifications happen after translation as well. So if you've ever wondered why we only have about 25,000 genes but can produce a lot more proteins, part of it is you can have post-translational modifications change the protein once it's been translated. So some proteins can fold on their own into their proper tertiary structure. Others need a chaperone protein to help out. And then you can also modify these proteins after translation, adding on different groups, cleaving or breaking the protein into multiple pieces. And all of these change the structure, which can then change the function. And then in eukaryotes, this tends to happen in the endoplasmic reticulum and also in the Golgi as well. Now, there is an exception to the central dogma of genetics. If you remember, we learned that information flows from DNA to RNA to protein. And in living things, this is always the case. The exception to this is viruses. There are some viruses called retroviruses, retro meaning backwards, where you actually have information flow from RNA to DNA. There are some very famous retroviruses like HIV is a retrovirus, COVID-19 is also an RNA virus, and so it's important to learn how these viruses work so that way you can also sometimes target therapies to treat these viruses before they can have a massive impact on somebody's health. So what happens is these viruses will contain an RNA genome, and they'll also inject an enzyme called reverse transcriptase into their host cell along with their genetic material. The job of reverse transcriptase is to do literally what the name says in reverse transcription. So instead of going from a DNA to RNA, you start with RNA, and reverse transcriptase makes a DNA copy of it. That DNA copy of the viral genome then gets integrated into the host cell's DNA, and then that allows the virus to basically replicate, to direct the host cell's machinery to start making new viruses. So a very common focus of therapy for some of these RNA viruses, these retroviruses, is to try and target reverse transcriptase, since it's not really an enzyme that eukaryotic cells like ours tend to make naturally. All right, that's it. Next up, we'll talk about how we control gene expression and get into some more complicated stuff.